This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Okay, who's going to record? Okay.
Good job.
Remember to do that. So I'll just start with the acknowledgement to country. So I acknowledge the traditional owners of the land on which we are meeting. I pay my respects to their elders past and present and the elders from other communities who may be here today, and acknowledge that sovereignty was never ceded. We're here today for Alice to do her confirmation presentation in her Masters, and Alice has joined us. She's one of the most organised students I think any of us have had, so we've been impressed by Alice from word go. But she's here with us on an Australia Awards scholarship from Indonesia and comes from Gadjah Mada. Well, did her DVM at Gadjah Mada and also worked on the COVID response. And I had all these ideas to getting her to work on our COVID project, but then she started talking to a colleague of ours, Tom Weaver, who was very keen on AMR in wildlife. And before I knew it, the project had changed under my toes. So Alice has basically come with her own ideas and projects to tack onto some work that we had going at Sasha's PhD project that we're also working on. So these things are all linked. I'll leave it to you, Alice, to take over, and good luck with your presentation.
Okay, thank you, Simon, for the introduction. And I also would like to express my greetings and gratitude to all of the participants who joined us today, 5 days before Christmas. So before we start, I would like to tell you about why instead of what. So I'll tell the why story behind this research. So during my previous work, I randomly said yes to my boss to attend a meeting that is totally unrelated to my work in an hour before the meeting took place. And the meeting appeared to be about establishing a national decree for antimicrobial resistance, or AMR, for AMR working group at the national level. And in this meeting, I learned from various stakeholders, institutions, and organizations that most of the AMR funding related to animal health goes to either poultry or food safety. And then my first reaction was, why? I mean, I know that we have AMR in wildlife too. Well, at least in Indonesia, I know that we have resistant bacteria in orangutan that is closely related to human. So then why we only focus on poultry or food safety? And hence my research, I'm trying to investigate the current situation of resistant bacteria of public health concern in wildlife, especially reptiles in Australia. Okay. And before we started, I would like to break today's explanation down into 5 sections, starting from introduction, and then my thesis chapter, and then how I design my study, how will I process the data, and my timeline. It will take around 30 to 40 minutes to finish the overall explanation, so just stay with me. And as for the introduction or background, I would like to begin with these 3 main ideas before we jump to my aim and scope. Okay, let's get started with the first main idea. What is antimicrobial resistance, and why is it an interlaced hazard to human health? There are countless of definition of antimicrobial resistance available, but this one is my favorite. So quoting from a book titled Antimicrobial Resistance in the 21st Century, AMR is defined as the resistance of microorganisms to an antimicrobial agent to which they were at first sensitive. I found that this phrase here, highlighted in yellow, guide me to understand the differences between what is actually resistant and what is actually not a resistant as I dug deeper into the issue. For example, some of you may be familiar with broad-spectrum antibiotics. That is a drug that can treat bacterial infection from various species. And on the other hand, we also have narrow-spectrum antibiotics, which only effective for few species of bacteria. Does it mean that the bacteria that cannot be killed by a particular narrow-spectrum antibiotics is resistant against them? Well, the answer is no, and the answer is the result of the 18th century debate about what is acquired resistant and what is natural or intrinsic resistant. And then in the 19th century, the debate is mostly about polymorphic or monomorphic resistant. And then in today's era, the debate is about adaptive and mutation, and they also have adaptative mutation somehow, and it's getting more and more confusing, at least to me. So having this definition in mind helped me to understand more advanced and more complex explanations so far. And I hope this definition will help you too to understand my next explanation. Moving on to how does the resistance even exist? So this slide is my summary from much information I gathered from the literatures. And I would like you to note that the literature may not describe all of these things all together, and they are not necessarily explained as a sequence like this or a process like this, but rather explained or described in a section. So the resistant bacteria can has the resistance itself at the genetic level, and this alteration in the gene can be resulted from mutation or acquired from other bacteria that is already resistant through vertical or horizontal transfer. I have some slides about this, but since I have limited time today, I will skip that for now. And bacteria that has resistance in their gene may express their resistance when challenged against a particular antibiotics. And this manifestation of the resistance is called phenotypic resistance. So for example, you have bacteria and you stick it on a plate and you put antibiotic on it and then you let it grow and there is no inhibitory zone. It means that the bacteria is phenotypically resistant. Not all of the bacteria that has genetic resistance is manifesting their phenotypic resistance. And once there are resistant bacteria among sensitive bacteria, a Darwin natural selection may occur. So how is that happened? Well, susceptible bacteria will die in presence of antibiotics and leaving these resistant ones to exist and become more and more dominant over time. And at the end, we have this population of mainly resistant bacteria. And now that we understand what is AMR truly is it really a problem? Well, there are past, present, and future data that prove that AMR is not a distant peril. In 2019, there were 2 million died attributed to AMR, and there is also data about resistant bacteria obtained from international traveling. I really like this data because it shifts your perspective from local or regional issue into a global matters. And currently, AMR is one of the top 10 global public health threats facing humanity declared by the World Health Organization. So this decision is rather justifiable, especially considering the prospected future burden of AMR. For example, by 2050, it is estimated that there will be 10 million people die in a year with 100 trillion USD burden and the GDP annual reduction as worse as 2008-2009 global financial crisis. You can also see here in the picture that AMR burden is even higher than cancer and many other diseases combined. So we are now moving to the second main idea. Previously, many people, including myself, think that AMR is a man-made phenomenon. However, recent research reported the existence of resistant bacteria in permafrost, that is a completely frozen area, and in an ancient cave without history of human contact at all. This discovery demonstrates that resistant bacteria has been here even before the discovery of antibiotics. Nonetheless, the emergence of resistant bacteria is getting more and more faster due to the massive use and sometimes misuse of antibiotics. So why something that is previously not a problem is now a problem? How do we manage so far? Previously, we are fortunate because every time we have resistant bacteria, we can create new compound or new agent to combat the bacteria. However, in the past 30 years, we experienced this antimicrobial discovery gap or antimicrobial discovery void, where there is no new substance even discovered. So currently, it is more harder to treat bacterial infection caused by resistant agent, especially if the bacteria is resistant against multiple antibiotics or multi-drug resistant bacteria. And this situation is attributable to the outrage of antimicrobial discovery phase. So as you may see here, it takes very long time with tremendous cost and very tiny probability of success, especially during these initial stages, or in this graph, they call it Valley of Death. However, putting this into perspective, I would like to ask a question. Let's say that we can create new antibiotics every time bacteria become resistant, just like the old time. Do you think that this kind of solution is the correct answer? Well, to answer that, I believe that we have to dive deeper into the issue, the root of AMR. And apparently AMR is not a mere pharmacological or microbiological issue, driven by these human influences on health, including societal foundation, economic, cultural, political, and technological issues that push our ecological and planetary boundaries, creating challenges in human health, environmental health, and animal health. So it is clear that for such a complex issue, creating a new antibiotics is just a temporary remedy. So what is the most sustainable solution for this? Current global consensus agrees that One Health is one of the most promising answers. What is One Health? Well, One Health is an approach that recognizes and aims for the balance of these three interconnected health, that is animal health, ecosystem health, and human health, through multi-sectoral 4C. As you can see here, it's communication, collaboration, capacity building, and coordination. So in short, it's just like people working together to achieve this thing. And we are moving to the next and the last main ideas. So we know that AMR is a complex problem and One Health is the most probable solution. And the question is, where is wildlife among those One Health constellations? And the most important question will be, why is it important for wildlife to be included there? From AMR perspective, the first reason will be because the resistant bacteria can spread anywhere. So for example, here, if we have resistant bacteria from wildlife, it can contaminate the environment and it can spread to aquaculture, food animals, other animals, and even to humans. And vice versa, resistant bacteria from human can spread to environment, wildlife, and other sentinels. And it's the same no matter where you start. And the most important thing that we have already learned previously is the resistant genes can be transferred anyway to other bacteria harboring in any host. So not only that the resistance can be spread, the resistant genes can also be jumped to other bacteria. But Alice, we like your story, we are now focusing on food animals and poultry because of its huge impact to human health. What is wildlife impact on human health? As long as we washing hands, we practice good hygiene, we handle reptiles with care, why bother to care about AMR in wildlife at all? Well, I disagree with that statement because at bigger perspective, wildlife plays important role in the ecology, in the environment, and thus important for human existence. Especially for reptiles, according to IUCN, 19% of these creatures are already threatened, and 21% others are classified as having insufficient data. However, reptiles play important roles in our existence. For example, they play as gene transporter, so they distribute the seed of the plant. Well, previously this idea is doubted, but recent research suggests the effectiveness of gene transportation by reptiles, especially since they don't digest the seed and thus eject the seed as a whole. And the next role that they play is nutrient transporter. So reptiles laying their eggs and give themselves as a prey, as a food for other animals. And the last one is trophic actors. So some of the reptiles are competent predators, and the shift in their population can flip the balance of the nature. So it's better if we can preserve the population of the reptiles from resistant bacteria as well. And the second reason is because the antimicrobial use in wildlife is trickier compared to other animals. We experience inadequate data because they are ectotherm, so their temperature is highly affected by the environment, and thus it is hard to measure the activity and the drug level in their body. Nowadays, we don't have any available guideline up to in related with antimicrobial use in reptiles. Well, an attempt was made by British Small Animal Veterinary Association or BVSc, but they just published suggestion on how to use antibiotics in reptiles based on previous case report. Therefore, veterinarians often extrapolate the dosage of antibiotic use in reptiles from other species, usually mammals, and often the antibiotics is given without proper diagnosis because, hey, the reptiles is harboring weird organism and we don't have adequate data about that as well. And the last one is some of the diseases in reptiles require broad spectrum antibiotics anyway. So yes, the irony. And in line with my opening story and as well as this result that I will talk to you later, wildlife focus in especially in AMR is still inferior compared to other sectors. For example, we have this organization here called quadripartite, consists of WHO, FAO, UNEP, and WOA, previously OIE. They publish a lot of documents about AMR. But you can see that here the WHO have this foodborne bacteria and FAO have this food animals for consumption AMR related thingy, but they don't talk about wildlife. Well, it makes sense because it's WHO and FAO, they don't care about wildlife. But we have here UNEP and WOA. So previously, some people categorize wildlife in environment sectors and others categorize wildlife in animal sectors. So it can be in the both sectors. Even in UNEP and OIE documents, there are no specific focus about AMR in wildlife. Well, OIE recently published many documents during previous World Antimicrobial Awareness Week, such as for farmer, for veterinary authorities, but there is none about wildlife, even in this year. And thus, my research is trying to fill the gap just a little. I'm aiming to identify the prevalence of the resistant bacteria in reptiles, including its risk factors, and hopefully I can identify the intervention point or appropriate strategy to combat AMR from these sectors. And then hopefully this data can be an insight for AMR arrangement, initiative, guidance, and regulation, especially here in Australia. And the scope of my study is prevalence study in Australia, targeting resistant bacteria of public health concern, which I will explain later. In healthy squamata consists of snake and lizard in different settings, so captive and wild and ecoregion, which I will explain real soon. And moving on to the next section is my thesis chapter. So because this is just a master research, so it's basically just one PhD chapter, starting from introduction, lit review, methods, results, and discussion. So for introduction and lit review, I explained some of them just now, and then I'll explain my method later. And then for result and discussion, it is mostly about prevalence study and cross-sectional study. Moving on to the next section, how I design my study. So I'll start with the helicopter view of my research, and then where the research will be conducted, and then the target bacteria, and then causal graph, and how I calculate my sample size. So overall, I will take swab samples from reptiles, from oral and cloaca, and I will also conduct a questionnaire study. So I'll ask the reptile owners or reptile carers to fill this questionnaire, consists of questions related to AMR in reptiles. And hopefully I can generate AMR prevalence in Australia, including its following risk factors. And this study will take place, well, this is kind of like tricky because it's not in a state base, but ecoregion base. So based on WWF, Australia has 7 ecoregions. And as you may see here, there are 2 tiny ones, and I will exclude that ecoregion because it will be unlikely to get adequate data from that. So I will focus on 5 largest ecoregions. And since I only have 2 years to finish this project, I'll start from one ecoregion first, trying to finish that, and moving on to the other ecoregion. So I plan to start from these ecoregions, which consists of Tasmania, Victoria, a bit of New South Wales, and up to Queensland. And how do I select my target bacteria? Well, I want this project not to be focusing in clinical related diseases in reptiles, but rather public health perspective. So I start with WHO AMR priority pathogens, which consists of critical, high, and medium bacteria. And also WHO vaccine availability. We have Group A, Group B, Group C, and Group D. So Group A means there are vaccines widely available. Group B and Group C means there are vaccines under development with different feasibility. And for Group D means there are no vaccine available and no vaccine under development and nothing. Why is this important? Because this ABCD characteristic define what is the most appropriate preventative strategies to combat AMR. For example, if you are dealing with Group A bacteria, vaccination will be the best approach. But if we are dealing with Group D bacteria, we don't have any vaccine, so preventative measure will be the best. And interestingly, most of the bacteria and reptiles belong to the Group D. So we have no vaccine for that yet. And since I start from human pathogens, I will also look whether this bacteria can exist in reptiles, especially snakes and lizards, and whether it is zoonotic and whether it can be detected by oral cloaca swab. And the last one is risk. So there is infection risk and resistant risk. Infection means a bacteria that can cause nasty disease, severe disease, but not necessarily resistant. But resistant risk mean bacteria that might be commensal, like natural bacteria that is opportunistically pathogen, but once it becomes resistant or moreover multi-drug resistant and somehow infect the host, it will be harder to treat. And those from these 6 criteria, I have this 10 list of bacteria, including Enterobacteriaceae, Enterococcus, and Staphylococcus aureus. And you can see here, I have this resistant, like ESBL, carbapenem, vancomycin, and methicillin resistant. These are the resistant concern of public health perspective. So I will only screen for this resistant, but I will do further sensitivity tests against many other antibiotics. And how I do my sampling. So my population is snake and lizard here in Australia, and my study population is captive and wild reptiles. And I will do 2-stage clustered sampling. For example, we have ecoregion A here in one ecoregion. I will have primary sampling unit that is a site defined by an address, either GPS location or postal code. And I will select sites within this ecoregion by convenience sampling. So it's a voluntary basis. And I will have this secondary sampling unit that is individual reptiles, either lizard or snake. And I will take samples from each site using a simple random sampling. And how much do I need to sample? I will need 31 primary sampling unit or sites and 62 secondary sampling unit or reptiles. And this number is for each either lizard or snake, or either captive or wild. So for example, for lizards, I will need 62 captive lizards in 31 sites. So multiplying with others, in total, I will need 124 sites and 248 reptiles in a ecoregion. And how do I get this number? Well, I start with the estimation of the prevalence of the disease in that population. So for AMR, it is quite tricky because I deal with not only one disease, not only one bacteria. So for Salmonella, the prevalence will be around 30 to 50 for captive reptiles and about 20 for wild reptiles. And for other bacteria, we don't know. So I estimated there will be 0.1 prevalence in the population, and I would like to be 0.1 accurate on this research with 95 confidence level. And this primary sampling unit, the population site, is defined by the number of reptiles license holder in each state. So it is estimated that will be around 6,000 of reptiles license holder in a state, and hence in Australia there will be 36,000 reptile license holder. But any number above 1,000 will not change the calculation anyway. And then I will take 2 samples in a cluster or a site, and within cluster variance of 0.2, it means that there will be little differences in a site because of the similarity of husbandry, biosecurity, and many other. But the between cluster variance is higher because from one side to other side, there will be huge differences in husbandry and biosecurity and many other more, and hence the variance is greater. And using this magic created by the EPI team, so Mark Stevenson and my supervisor, Simon Firestone, I get this number. The required primary sampling unit is 31 and the required secondary sampling unit is 62. And I also create this deck or causal graph. So many people may not be familiar with this. So this is the idea behind this is to seek what is probably the cause of the outcome of the research. So for example, here my outcome will be reptiles with resistant bacteria. So what might be affected this outcome? The first one is antimicrobial use in reptiles. And the second one is reptile source of food, because some literature mentioned that mice might lead to salmonella outbreaks in reptiles. And then biosecurity breach, which closely related with reptile carrier, if whether they are harboring resistant bacteria or not. And then the aforementioned ecoregion, and then reptile signalment is just human demographic, so age, sex, breed, and then husbandry, how the owner keep the reptiles, such as temperature map, UV wavelength, because reptiles are highly affected by those. And the last one is animal use or care occupation, because some reptiles might be used for educational purpose and thus change the frequency of human contacts. And this for down below affect the microbiome of the reptiles and will eventually affect my outcome, that is reptiles with resistant bacteria. And this causal graph is not only useful to analyze the data later, but also to design my questionnaire to make sure that I don't miss any important information that I need. And moving on to the next section is my research method. So how will I collect the sample? How will I isolate and identify the bacteria? How about my questionnaire? How will I analyze the data? And how will I manage the data? So for sample collection and microbiological procedures, it will be conducted under the Australian Code for the Care and the Use of Animals for Scientific Purposes, and also the Animal Ethics ID 24121, which is already approved. And the owner will also require to sign a written informed consent before having their animal sample. So reptiles with active bright alert more than 100 grams and non-venomous will be handled and restrained in accordance with British Small Animal Veterinary Association Manual of Reptiles, and a swab, one from oral and one from cloaca, will be taken and cultured in less than 24 hours in sea blood agar and McConkie agar. And then it will be subculture until it reaches pure culture. And after the bacteria can be identified, it will undergo a sensitivity test using sensitizer. And for the questionnaire, it will be conducted under the UNIMEL Human Ethics ID 25458, which is under review. And the participant will also require to tick their consent through the online questionnaire. And this is based from the previous causal graph. So from that causal graph, I identify what might be the drivers, and I create several questions related to those, and I categorize them into these 4 sections: contact, biosecurity, husbandry, and human. And using this Qualtrics, it's an online survey tools, I distribute these 4 categories into sections because not everyone has to answer the question, so I will need a skip logic for the questionnaire, and also to make sure that the questionnaire can be understood easily by the participant. And for the data analysis, for the prevalence study, I will use basic epidemiology, and then for the cross-sectional study, it will generate odds ratio, and it will be adjusted by regression multivariable analysis using the previous deck. And for the data management, I'll collect a lot of data, like physical data like swab specimens, and then digital data, photos, MIC results from the computer, and then all the data will be anonymized and their access will be restricted only to the researcher, and it will be stored mainly in the university server with secondary storage in laptop and tertiary storage in external drive with backup every day. And moving to the next section, the timeline. So it all starts with this, the microbiological procedure, because it's defined how long I will process each sample, and then my overall timeline. For the microbiological procedure, this is the longest and the safest timeline because I know that bacteria from reptiles are weird, and I am not an expert in microbiology, so I need time to identify the bacteria. So the first week aim is to reach pure culture, and then week 2 aims to identify the bacteria, and after it can be identified, I will do the MIC test using sensitizer. And this is my overall timeline. You might like to focus on this red rectangle here. So I finished most of my induction and course and trainings, and also the ethics is the animal one is approved and the human one is under review. So in the next 6 months, I'll mostly do my sample collection and also simultaneously do my writing because my thesis is a living document anyway. And this is the last section. So closing, I would like to thank the University of Melbourne and also Australia Awards that grant me this scholarship to finish my master degree. And this Australia Award is funded by Australia Department of Foreign Affairs and Trade, or DFAT, and then SciPac Foundation for funding this project, this research project, and then my supervisor, co-supervisors, advisor, research partner, and the lab technical officer. And thank you for your attention. I'll take any question.
Well done, Alice, and kept the time really well. 36 plus 1, so that's wonderful. Well, we have a first question, Franciscus, who is now in the research group at Werribee, that was former zoo vet. I knew him at Hillsville, but has asked about the selection of non-venomous reptiles. So I'm not happy for you to come on, Franciscus, if you want to put your camera on and ask a question, or we can ask it for you. Yeah, so Oh, here we go.
Hi, great job. Great talk. My question was just around, is there the potential for bias if you're not going to sample venomous reptiles, because you won't get a good representation across all the ecoregions? For example, in Victoria, any snake you're going to get is going to be venomous. So basically means you won't be sampling a whole, quite a large cohort of animals from this state.
Okay, well, yeah, it's one of the limitation of this study, because for wild sampling, I will work with researcher in ecology, so they will have this license to handle venomous snakes, and I don't have that license, and I will not be able to coordinate with all of the people that hold that license, so it will be one of the limitation of the study. But I have that number here. The 62 captive and wild. So for the wild one is 62. I'm aiming to get the 62. If I can get the venomous one, then it would be okay. But I'm not expecting to get that much. So yeah, the representativeness and from the venomous animal will be the limitation of this study. But if you have like any contact information or collaborators that can handle these venomous animals and have the license that makes me able to collect the samples, I will be very grateful, because it will be like more representative. The study will be more representative.
If you want to contact me off outside of this this presentation, I can potentially give you some tips.
Yeah, thank you, Franciscus.
No worries.
And Sasha's just reminding us that we're sampling venomous snakes, but only cloacal. So I think so I think we need to be aware of that, definitely in areas where it might cut the numbers down. But we might. The next question is some challenging questions. This one will be hard, Alice, because you've only been in the country for 4 or 5 months. But Shitiz is asking a question about reptile farms in Australia. I don't know if you want to ask it or you want me to ask for you, Shitiz. I can ask for you. Yep. So he's asking, are there any reptile farms in Australia that you're sampling from? There are farms in Asian countries and the Philippines. Would there be AMR and reptile farms in the Philippines?
Thank you, Citis, for the question. So yeah, because as you can see on the map, my location is quite weird. It's not a state, it's ecoregion, and I will be working not by myself, but with many other collaborators out there. So I believe there will be, I don't know if reptile farm is the correct definition for captive reptiles, like for breeding purpose, but I expect there will be that kind of farm or breeding site in Australia. But for Asian countries and Philippines, well, from my lit review, I found that data about AMR in reptiles is very inadequate. So mostly it's about reptiles that are kept as pet animals. So they gather the data from clinics and animal hospitals. So for farms, I haven't seen one during my lit review. So I don't know whether there will be AMR in Philippines or even my country. Yeah, even in Australia, I don't know. I don't think I have that kind of data yet. I think
Alistair's commented on what I would know about the crocodile farms, of course, up in the north, but we're not targeting. Yeah, we're only targeting lizards and snakes, and I'm really not aware of any snake farms as such. But there may well be. I suppose, yeah, an interesting thing is if we are sampling from them, then we have to think about how many samples we take from a certain site, and and we thought about exhibits in zoos as sites within sites. So the same thing would apply to farms. But yeah, it's not been something we thought a lot about. Yeah. Yeah. So next, sorry, just before James's question, Nadika's had her hand up, so I might just ask her to ask her question first.
Thanks, Simon. Thanks, Alice. It's a great talk. Very interesting project. My question is, so you had a very nice figure that shows what would affect the presence of MR bacteria in captive breeds, captive animals. So when you are analyzing the data from the wild animals, wild snakes and lizards, would you be using a similar figure, or would you be using something different, because the effect, the things that affect the wild animals would be different, isn't it? Yeah, that one.
Okay, thank you, Nadika. Am I pronouncing your name correctly?
Yeah.
Sorry if I'm wrong. Yeah, thank you for the excellent question. So for these drivers of MR in wildlife and captive ones will be much or less different, but mostly it's just the same. For example, here in reptile signalement, for the captive reptiles, because the owner can keep anything, like there is no rules, they have to keep something that match their ecoregion. So the reptile signalement will not be affected by ecoregion, but in the wild one, the ecoregion will affect the species in that area. So there will be an arrow from ecoregion to reptile signalement. And for other things, such as husbandry, it will be hard because they are in the wild. They don't be taken care of by anyone else. That's why the question in the questionnaire is different. So for wild reptiles, they only ask about the biosecurity because, well, even I don't expect that the reptile carer will be remembering everything about this particular reptiles, especially if they are wild, just being caught. We cannot identify whether this one is the previously treated or something like that. So yeah, there will be differences of the data of the AMR drivers between captive and wild one.
Yeah, thank you.
Thanks, Nadika. Okay, James on his iPhone has asked, is there a concern that multiple subcultures prior to AST will allow isolates to lose their resistance plasmids and potentially change their results?
Yes, that is interesting because that is the debate about, is it polymorphic and monomorphic resistance? So the idea whether the resistance can be retained in the bacteria. So yeah, I'm not planning. I'm actually right now doing the pilot project, James. Like I have several samples and I process some. I don't know how long with my like current skills and knowledge to isolate from the primary isolation to identification and to antimicrobial sensitivity test. So I will look at that, and probably I can try to make longer subculture to make sure whether there will be potential change on the results if I subculture it many times. Like it will be interesting to see during my pilot project.
As we've also got a lot on your plate there, Alex.
Yeah.
We were talking at the start about each sample you take multiplying into multiple more. So we have a follow-up project planned that will involve some of the sequence work, and so I think Alice's focus will primarily be on the phenotypic aspects and the culturing. Yeah, for the PhD, it was only so much we can pack into a master's. We're already trying to scope it down. And Sasha's mentioned that snake and lizard breeders for the pet trade will be part of those that are sampled. Yeah, so this comes into how many we take from each side. Steve from WDA, Wildlife Disease Australia, I think it's mostly a comment. I couldn't see a question there. I know, just
yes.
have a question there. Are there any possible research links with general microbiome knowledge in reptiles, e.g. AMR bogs more likely in herps, which I suppose are frogs, with reduced microbiome diversity? And any insight on bacteriophages, both as pathogen and treatment in AMR research in reptiles, especially those kept as pets? Probably a question that Mark...
Bogs, more likely
Mark
herps.
Yeah.
Okay, so yep. Hi, Steve. Nice to meet you again. And is Steve still here? And yeah, interesting question. And I don't think I have adequate information to answer this question now, but I can say that I have this slide here. Wait. Which one? So it's this one. So during the selection of my bacterial target, oh, I cannot share that one. During the selection of my bacterial target, I realized that there are differences of microbiomes, obviously, between species in reptiles. So, for example, here I'm trying to target this one, Shigella, but I decided not to because most of the bacteria is in turtles instead of snake and lizards. So definitely there are diversity of microbiomes in AMR, but I don't know if it is necessarily to be related with the resistance itself. This one is just the microbiome of or the proportion of the bacteria or the prevalence of the bacteria, but not the resistance itself. And any insight on bacteria facts? I haven't found anything about this yet. So yeah, I cannot answer that particular question right now. I haven't seen any literature about that. Yes.
That's definitely PhD number 3 or postdoc. So yeah, we'll get to that one later, I think. Okay, Alistair, I'm not sure if this question, metagenomic sequencing to detect for bacteriophages, yeah, is a PhD beyond that. So I think there's lots of avenues further on. All right, we'll just, I think, gosh, we've given you a lot there and you've covered a lot of ground, Alice, and definitely shown your knowledge and how much you've learnt and read in the last, it's only been 5 odd months since you've been with us. So that's a fantastic effort today. If anyone's got any further questions, throw your hand up now, or we'll let Alice off the hook for the day. But I think a magnificent presentation. It shows how much you've learnt already and how planned you are coming ahead for your study. Thanks for some of those questions, Franciscus. That's really helpful. We'll think more about the venomous aspect and discuss this, Sasha, and we'll probably get back to you. So as soon as we have plenty of ideas for the PhD, postdoc, and career beyond it. So thank you everyone for attending and and for your attention.
Thank you everyone.